Today I'm going to be investigating and talking about polymorphs and co-crystals. So the traditional approach to solid form screening is pipetting two solutions into a microwell plate. They also have automated systems that can perform this on a high throughput basis. Our approach is using a microfluidic platform, which will still maintain the high throughputness of this process while consuming minimal amounts of sample. Additionally, we want the process to be easy to use, and so for our microfluidic device, all you need is a pipette and a vacuum pump, or some source of vacuum. The device fabricated in our lab has two layers. There's the control layer, which is used for fluid routing, and the fluid layer, which is used for fluid storage. In this blown up schematic over here, you can see that one compartment will hold our active pharmaceutical ingredient, while the other will hold a counter ion, so in the case of a co-crystal screen. We also have two sets of valves, one for filling in the chip and one for mixing the two solutions together. The valves are normally closed, and at the bottom here you can see this little diagram showing that once vacuum is applied, this membrane in the middle will tense up and the two solutions will be allowed to mix. So what you're seeing here is basically just a cross section through this center well. Now I'll show a short video of how this chip actually operates. So first, the valves are actuated. You can see they turned a little bit dark, and the fluid is being filled. What's actually happening is over here, we're pipetting a droplet of solution onto an inlet, and over here, we're pulling vacuum. So this will allow our chip to fill easily, and we'll be able to screen a lot of conditions. After the chip is filled, the valves are closed, and the mixing valves in the center will be actuated, allowing for diffusion to occur. So in this process, or in this chip, we're using two different materials. Traditional microfluidic material, PDMS, which is used for the valving application, and we're also using cyclic olefin copolymer, which is used as a substrate on the top and bottom of the device. This COC is solvent impermeable, which reduces the solvent escape through the PDMS. The wells here hold volumes of 50 to 100 nanoliters, and this system is compatible with a wide range of solvents, including polar organic solvents such as alcohols and water and acetonitrile. Additionally, because the valves are normally closed, this chip is portable, and we can do on-chip analysis. So a previous member in the group validated this platform. He looked at caffeine as the active pharmaceutical ingredient, and he did a co-crystal screen. So in the first chamber, he filled in caffeine in a solution, and in the second chamber, he filled in 2-hydroxy-1-naphthalate, and in the second, he filled in 3-hydroxy-2-naphthalate. And after a few days, he saw crystallization. He did Raman to identify the solid forms. At the bottom, you see that the chip does have a Raman pattern, or does have a Raman fingerprint, but it doesn't seem to get in the way with the key peaks for caffeine, which is nice. What's interesting though, or the challenge that we're having, is that sometimes it's difficult to determine exactly which co-crystal we're getting. So sometimes it can, or the spectrum between the two co-crystals is very similar. So what we'd prefer to do is be able to definitively tell which co-crystal we're getting. And some cases we want to know the complete crystal structure. Raman just will be able to tell us that we have a different solid form. We want to know exactly what that solid form is. So my goal was to perform on-chip analysis using X-ray crystallography. So the first question is, are these chips compatible with X-ray? So previous members in our group are working on protein crystallization and trying to determine the 3D structure of proteins. They have a similar chip in the sense that they're using COC and PDMS, and when they've collected diffraction data, they see that there's two concentric rings over here, which is the background from the chip. They found that this does not get in the way of the scattering of their crystals, which is fortunate. Additionally, to make our chips integrate well with traditional X-ray sources, we needed to develop a new crystal mount, or a new way to mount our crystals. So down here we have a traditional crystal mount. So up here we'll have a loop where traditionally there'd be one crystal attached. Here we have a mount that our entire microfluidic device can be placed into. This little screw will hold it in place. And here you can see that this chip has been mounted on the Green Line at Argonne National Labs. So the next problem is that when we do these experiments, we get crystals that tend to look like this. So they're not very good quality crystals, and they're not separated. So what happens during our experiments is once the solvent's evaporating, we reach a critical supersaturation ratio where we induce primary nucleation. Ideally, we'd like this to fall down into the growth phase, and we'll grow a certain number of crystals. What tends to happen is we fall into the secondary nucleation phase and we just get a scatter of crystals. My approach, I'm interested in doing a seeding approach, which is a common approach done in crystallization, where you introduce a tiny crystalline particle, and instead of having to reach such a high level of supersaturation to reach nucleation, we can just reach a lower level of supersaturation and induce crystal growth. So the idea is to decouple crystal nucleation and growth. The general approach I'm using is I'll crystallize the drug or the active pharmaceutical ingredient off chip. Here you can see in these vials, this is just an evaporation experiment, kind of like the traditional approach. So we crystallize the solid. They're not very good crystals, but this is what we're going to use as our seeds. We do Raman to characterize that we are indeed getting the crystal form that we want. We'll then crush these seeds in the saturated API solution so that the seeds don't dissolve. Once we have a stock of the seed solution, we will dilute the solution, and the idea is that the diluted solution will give fewer crystals and the crystals will be larger. Once we have this diluted solution, we introduce it on chip. So in the top ratio, or this ratio is referring to the seed dilution previously shown. So in this top chamber, we'll have seeds, so small crystals in the saturated drug solution. In the bottom chamber down here, we'll have our active pharmaceutical ingredient and counter ion. We'll activate the mixing valve in the middle, allow 5 minutes for diffusion to happen, and then we will let the solvent evaporate, and in about a day, we should see good quality crystals. The compound I chose to use in this case is carbamazepine. I chose it because it's been well studied, and there are crystal structures reported for both of these compounds. So I'm looking at carbamazepine by itself, so crystallizing form 3 of carbamazepine. One of the reasons I chose this compound also is because it's known to exhibit different polymorphs. So carbamazepine by itself will have at least 4 crystal structures. And carbamazepine in the presence of different acids will form a cocrystal. And the cocrystal I'm interested in is 4-hydroxybenzoic acid. Carbamazepine is commonly used in the treatment of epilepsy and bipolar disorder. So once I carried out these seeding experiments, we found that indeed the fewer crystals that we introduce on chip, we do see better quality crystals, and they do tend to be larger on average. You can see that the control did not yield very many good crystals. Occasionally, we did see crystals that would be diffraction quality, but on average, we did not. And also in the case of carbamazepine and 4-hydroxybenzoic acid, we saw the trend also that the more dilute solutions give larger, fewer crystals. You can see in the control that we do see some good crystals also, and this wasn't a surprise. I chose this crystal system because it forms prismatic crystals, and so in this case, this approach wouldn't necessarily be needed, but we just wanted to verify that our seeding approach would work. So one assumption that we're going to make is that all of these seeds are randomly oriented, or all of these crystals are randomly oriented. So once we have our crystals on chip, we go to Argonne National Labs and use the advanced photon source to collect data. What we do is we bombard one crystal with X-ray and collect the diffraction pattern. When the crystal's in the chip, we can rotate it maybe 20 to 60 degrees depending on the size and orientation of the crystal. And because of a geometric constraint of our chip, we can't rotate it much further. So after we collect a certain number of degrees of data on one crystal, we move to the next crystal and again collect 20 to 60 degrees of data. And so it's important that the crystals are randomly oriented, because if they were in the same orientation, we would just be collecting the same data over and over again. After we've collected sufficient data, we come back and we'll merge all these data sets together in order to generate one electron density map and ultimately arrive at our final crystal structure. So we've been able to, in the case of carbamazepine and or HBA, we were able to determine the complete crystal structure up to 99% completeness, all with data collected on chip. So this is ultimately kind of the goal. So we see also that with previously reported data, the unit cell correlates quite well. We did collect our sample at room temperature instead of at the cryo-cooled temperature, so our unit cell is a little bit larger as expected. And so for these systems, the crystal structure has been reported. We didn't necessarily need to go through this entire process. But there are crystal systems, such as the case of carbamazepine and L-malic acid, where the crystals are very thin, they diffract poorly, and they're very fragile. So when we grow them off chip, we can't grow a large enough crystal to actually get the crystal structure. The co-crystal has been confirmed with Raman and powder X-ray, but no actual crystal structure has been able to be determined. So with our approach, we can keep the crystal in the microfluidic chip, so we don't need to harvest it. And what we can do is collect data from each of these maybe red lines. So we'll be able to get sufficient data to be able to merge it all together and then arrive at one coherent crystal structure. So in conclusion, I was able to grow diffraction quality crystals via the seeding approach, and I was able to validate that our microfluidic platform is X-ray compatible. I'm continuing to work on determining the structure for carbamazepine, and I'll still be collecting more data to determine the structure for carbamazepine and L-malic acid. With that, I would like to acknowledge Dr. Paul Kennis and my lab mates, also our funding source AbbVie and our collaborators at AbbVie, and the staff at the Argonne Beamline who helped with this project. And with that, thank you, and I'll take any questions.